Welcome, Arpan. Thank you for joining the session today. I'm excited to learn more about your background and assess your skills. To begin, could you please tell me about your academic background, including your relevant coursework and any projects you've worked on?
Yeah, sure. Yeah, sure. Hi. I'm a graduate student in biophysics, learning about potassium ion channels and how this potassium ion channels is regulated and structural and functional dynamics. Whatever goes on in human physiology, I try to mimic those in forms of liposomes and study.
Thank you for sharing that, Arpan. It sounds like you have a solid foundation in biophysics and a keen interest in understanding the complexities of human physiology through practical applications. To delve deeper, can you explain the basic structure and function of potassium ion channels? How do they contribute to cellular processes?
Yeah, sure. So potassium ion channels are basically membrane proteins and they act as a gatekeeper to pass selectively potassium ions. This is done by a particular sequence present called PVGYG motif in the selectivity filter. To say overall in yeast, it has an extracellular site, transmembrane region, and a cytosolic domain. So yeah, that's embedded in the membrane system and our cellular membrane and to maintain the potassium ion concentration inside and outside, like you know that inside potassium ion concentration is 150 millimolar and outside it's around 40 to 30 millimolar. So to maintain that balance, to maintain cellular homeostasis, these ion channels function. Yeah.
Thank you for your detailed explanation, Arpan. You've clearly articulated the structure and function of potassium ion channels, as well as their role in maintaining cellular homeostasis. Your understanding of the selectivity filter and the concentration gradients is commendable. Now, let's move on to the next question. Can you describe the process of action potential generation in neurons? What role do ion channels play in this process?
Yeah, before that, so how many questions do I have to answer in order to assess myself?
In this session, you will answer a total of 10 questions in each round. After completing each round, I will provide you with feedback on your performance. You can then decide if you wish to continue to the next round. Now, let's proceed with the current question. Can you describe the process of action potential generation in neurons and explain the role of ion channels in this process?
Yeah, sure. So what happens is the cell is at resting membrane potential at minus 70 millivolt, and then the potassium ions move out of the cell, creating a depolarization state, and simultaneously the sodium channels comes inside. So potassium ions are more outside and sodium ions are more inside. So after a particular time, there is a repolarization state, and then there is hyperpolarization state where a particular class of potassium ion channels called Kerr channels comes into play. These Kerr channels, potassium inward rectifier channels, again moves the potassium ion from outside to inside the cell to maintain the cellular homeostasis, and subsequently all the other channels function, and the cell gets back to the resting membrane potential to fire the next action potential, right? So what is the role of the ion channels in process? These ion channels trigger the course of action potential. Suppose you get your senses, your cardiac function, your kidney function, everything that's controlled by these ions moving in and out of the cell. And they bind to a particular target where they fire the physiological processes.
Thank you for your thorough response, Arpan. You've provided a clear overview of the action potential generation process, highlighting the roles of sodium and potassium ion channels effectively. Your explanation of depolarization and hyperpolarization, as well as the importance of ion movement in maintaining cellular homeostasis, demonstrates a solid understanding of the topic. For feedback, I would encourage you to focus on clarity and conciseness in your explanations. While your knowledge is strong, simplifying complex concepts can help ensure that your audience fully grasps the information. Now, would you like to continue to the next question?
Yeah, sure, I'd like to continue to the next question. Thank you for your feedback.
Great to hear that you'd like to continue, Arpan. Let's move on to the next question. Can you explain the concept of enzyme kinetics? Specifically, what is the Michaelis-Menten equation, and how does it relate to enzyme activity?
So what enzymes are, they are like catalysts, right? Enzymes remain unchanged at the end of the reaction. You have a reaction from A to B, and which takes a significantly longer amount of time, and you put in the enzyme, um, and the reaction is fast. So how fast the reaction happens is the enzyme kinetics, and this enzyme kinetics is dependent on substrate concentration and the reaction rate, and that is defined by the Michaelis-Menten equation, right? So yeah, Michaelis-Menten equation is nothing but a react equation which is involving the substrate concentration and the reaction rate.
Thank you for your explanation, Arpan. You've provided a good overview of enzyme kinetics and the role of enzymes as catalysts in biochemical reactions. Your mention of the relationship between substrate concentration and reaction rate is key to understanding enzyme activity. However, I would encourage you to elaborate a bit more on the Michaelis-Menten equation itself, including its components, such as Vmax and Km, and what they signify in terms of enzyme efficiency and affinity for the substrate. For feedback, I suggest focusing on providing specific details about the equation and its implications, as this will strengthen your understanding and communication of the concept. Shall we proceed to the next question?
Yeah, sure.
Thank you for your confirmation, Arpan. Let's move on to the next question. NN, can you discuss the differences between prokaryotic and eukaryotic cells? What are some key features that distinguish these two types of cells?
So prokaryotic cells are simple organism cells, right? They do not have membrane-bound organelles, whereas eukaryotic cells are complex that has evolved from the prokaryotic cell that has a membrane-bound organelles. Furthermore, prokaryotic cell lacks nucleus um, or a nucleolar organization, right? And a eukaryotic cells completely has a particular nuclear envelope and the nucleus embedded inside it. And yeah, so that's it.
Thank you for your response, Arpan. You've highlighted some important distinctions between prokaryotic and eukaryotic cells, particularly regarding the presence of membrane-bound organelles and the nucleus. Your understanding of the structural differences is clear. To enhance your answer, I would suggest mentioning additional features, such as the differences in size, the presence of cell walls in prokaryotes, and examples of each type of cell. This would provide a more comprehensive view of the topic. For feedback, I encourage you to elaborate on your points and provide specific examples to strengthen your explanations. Would you like to continue to the next question?
Yes.
Thank you for your willingness to continue, Arpan. Let's move on to the next question. NN, can you explain the concept of homeostasis? Why is it important for living organisms, and can you provide an example of a homeostatic process in the human body?
Yeah, sure. So homeostasis is particularly maintaining a balance, right? So you have your ions inside, like I said, the potassium ion concentration inside the cell is 150 millimolar, whereas outside it's 40. So whenever the potassium ion is irregularly balanced, the potassium ion channels comes into play, and it passes the potassium ion from outside to inside, or vice versa, wherever the concentration is higher. So that's homeostasis, right? Suppose you have a viral infection in your body, and you need to combat that. So a body increases the temperature so as to curb these viruses out, or any sort of infection out. So that's you get it in the form of fever, right? So that's homeostasis process in general. Anything that is irregularly controlled, you need to get back to the normal course. That's homeostasis physiology. Yes.
Thank you for your explanation, Arpan. You've effectively described the concept of homeostasis and provided a relevant example of how the body regulates potassium ion concentrations and temperature during an infection. Your understanding of the physiological processes involved in maintaining balance is commendable. For feedback, I would suggest being a bit more structured in your response. Clearly defining homeostasis at the beginning and then transitioning into your examples can help improve clarity. Additionally, mentioning other examples, such as blood glucose regulation or pH balance, could further enrich your answer. Now, would you like to proceed to the next question?
Yeah, but can I get a feedback now? Can we have to do 10 questions particularly, or can I get a feedback now?
Thank you for your question, Arpan. Yes, you can certainly receive feedback at this point. In our current format, we typically conduct 10 questions in a round, but I understand the importance of feedback for your learning process. So far, you have demonstrated a solid understanding of fundamental concepts in your field, and your ability to articulate your thoughts is commendable. Here are some key points of feedback. 1. Clarity and structure asterisk. While your knowledge is strong, focusing on structuring your answers more clearly can enhance communication. Start with a definition or overview, then provide examples or details. 2. Depth of explanation asterisk. In some responses, you could benefit from elaborating on key concepts, such as including specific components of equations or additional examples to illustrate your points. 3. Engagement with the question asterisk. Ensure that you directly address all parts of the question asked. This will help demonstrate your comprehensive understanding of the topic. Overall, you have shown great potential, and I encourage you to continue refining your communication skills. Would you like to continue with the next question, or do you have any other queries or topics you would like to discuss?
No, I'll continue later on, but thank you for your feedback. Thanks.
You're welcome, Arpan. I'm glad you found the feedback helpful. Whenever you're ready to continue or if you have any questions in the future, feel free to reach out. Thank you for your time today, and I wish you the best in your preparations. Have a great day.